think we're going to go ahead and get started so that we don't run into the next symposia, because I really want to give these young investigators plenty of time to present to you their outstanding work that led to their being selected as the for the New Investigator Awards. So first I want to introduce introduction for James Kokendorfer for this outstanding award, and what he's going to show you is a glimpse into his pioneering work in developing the concept of targeting the B-cell maturation antigen as a target antigen to treat multiple myeloma. In addition to that, he's developed an effective CAR T-cell incorporating this anti-BCMA antibody, including the preclinical work that validated this technology, and then conducting a clinical trial demonstrating the first in-human effective control of multiple myeloma with a CAR T-cell using this engineered system. And it's a real pleasure for me, and I'd like you to help me welcome James as he presents his work.
Good afternoon, and thanks to all you for coming, and especially...
You're a little taller than I am.
Okay. Good afternoon, and thank you all for coming, and especially thanks to the selection committee for selecting me for this very wonderful award. I greatly appreciate it. It's a great honor. And thanks for the wonderful introduction. So I'll go ahead and get started. My talk is going to be mainly a summary of all my chimeric antigen receptor work that I've been doing for the past several years. It's going to be, some of it will be the BCMA CAR. The first part is going to be a bit about the anti-CD19 CAR T-cell therapy for lymphoma. First, disclosures. I have cooperative research agreements with Kite Pharma and Bluebird Bio. I have multiple patent applications for CAR T-cell technologies. First of all, I know most of you are aware of this, but just to go over the different components of a CAR, this is an example of an anti-CD19 chimeric antigen receptor. I'm not sure. Is there a pointer? Where's this? Oh, sorry. Here we go. Sorry about that. So the various components, we have an antigen recognition domain, a T-cell here, various hinge and transmembrane regions are used to connect the antigen recognition domain to T-cell signaling domains that are in the cytoplasmic part of the T-cell once the CAR is expressed. So some type of vector, either a lentivirus usually or a gamma retrovirus, or in some cases a transposase system is used to transduce the T-cells, thereby giving the T-cell the ability to express the CAR on the T-cell surface. The T-cell then acquires the ability to specifically recognize a tumor antigen, in this case CD19. So for part one of my talk will be CAR T-cell therapies for lymphoma. And this work started back in 2007, which led to the first demonstration of antigen-specific anti-CD19 CAR T-cell activity in a human, which occurred in a lymphoma patient that we published in Blood in 2010. This was work conducted in collaboration with Steven Rosenberg when I was a postdoc in his lab. We had a patient with lymphoma who had eradication of normal B cells and a response with his lymphoma. He is right now still progression-free, 7 years after his last therapy, and living a completely healthy, normal life in Florida. We went on to show that CAR T-cells can be effective against diffuse large B-cell lymphoma, which is an especially treatment-resistant type of B-cell lymphoma. Along the way, we've constantly been guided by this finding that many investigators have shown in mice that chemotherapy administered before adoptive T-cell therapy dramatically enhances the activity of CAR T-cells and also other types of adoptive T-cell therapy. In some of our earlier work, we used fairly high doses of cyclophosphamide in some of our regimens. We reasoned that a conditioning regimen with a lower dose of chemotherapy would be less toxic and allow a more clear demonstration of the anti-malignancy activity of CAR T-cells against lymphoma. So we went on to test this regimen of a low dose of cyclophosphamide, 300 to 500 milligrams per meter squared for 3 days, given on days -5 to -3, and also along with fludarabine on days -5 through -3. Then on day 0, we gave a single infusion of CAR T-cells. This is not actually on in paper publication, but it's available online. What we saw with this chemotherapy was a very interesting change in multiple serum proteins. IL-15 went up in 22 out of 22 patients after this chemotherapy regimen. As you know, IL-15 is a very important T-cell active cytokine that can cause proliferation and activation of T-cells. IL-7 also went up. Macrophage chemotactic protein 1 went up in every patient, and perforin went down, perhaps as just a measure of lymphocyte depletion. So these findings are consistent with earlier studies done in mice by many investigators, and we were pleased to see that our regimen, which is a very non-toxic regimen that can be given as an outpatient, given to outpatients with very mild toxicity, was effective at modulating these serum cytokines. We went on to conduct our trial with this vector. This is a vector that was first published in Journal of Immunotherapy 2009. It has a murine single-chain FE called FMC63, CD28 hinge transmembrane and co-stimulatory domains, and a CD3 zeta T-cell activation domain. Our T-cell culture process is very simple. We just stimulate whole PBMC with the OKT3 anti-CD3 monoclonal antibody. The process takes only 6 to 9 days. We just stimulate the PBMC, let them transduce on day 2, let them grow to 6 to 9 days, and then prepare the cells for injection. So this trial enrolled patients with ECOG 0 or 1 performance status. We treated a total of 22 patients, 19 with diffuse large B-cell lymphoma. 11 of the 19 diffuse large B-cell lymphoma patients had chemo-refractory lymphoma. We also treated 2 patients with follicular lymphoma and 1 with mantle cell lymphoma. 7 patients received 1 million CAR T-cells per kilogram. 14 patients received 2 million CAR T-cells per kilogram. And 1 patient received 6 million CAR T-cells per kilogram. Here are the responses. Out of the 22 patients, overall response rate 73%, complete remission at 55%, PR, partial remission, and 18% for the non-oncologists in the crowd. Complete remission means there was no evidence of lymphoma, and partial remission means the lymphoma decreased by 50% in size. Among the diffuse large B-cell lymphoma patients, the overall response rate was 68% with complete remissions and 47%. Both follicular lymphoma patients obtained complete remissions, and 1 mantle cell patient received a complete remission. One of the important parts of this work is the duration. This and the durability of these responses. Out of the 12 complete remissions, 11 are still ongoing today, and the durations are here with plus signs indicating an ongoing response. They're ranging from 24 to 7 months. This is an example of one of the remissions. The red area here is lymphoma, indicated by the white arrow. This patient had lymphoma that progressed on 3 different very intense standard chemotherapy regimens. On the CAR T-cell study, her lymphoma was eradicated, and you can see here an absence of this red area, a cross-sectional cut of the same patient with the yellow area showing lymphoma, and with the disappearance of the lymphoma. This is a scan done 10 months later. She's now 18 months after treatment, still in complete remission. So this is an example of a patient that really had no treatment options. When you're refractory to all these chemotherapy agents, you really have nothing else that will be curative. Patients did experience significant toxicity. 12 out of the 22 patients, 55%, had grade 3 or 4 neurologic toxicity, including confusion, dysphagia, encephalopathy, and gait disturbances. This was by far the most prominent toxicity. We had only 4 of 22 with hypotension. This is in some contrast to the results with other types of diseases like leukemia, multiple myeloma, where disease burden in the body might be higher. I think the lymphoma patients are less prone to the hypotension type toxicities, but we certainly had a significant amount of neurologic toxicity. All toxicities resolved, usually within 2 weeks. We found some interesting correlative studies with this. We found an association between peak CAR-positive T-cell level and response. Those patients obtaining complete remission and partial remissions had a higher peak level of CAR T-cells per microliter than those patients who did not achieve a complete remission or partial remission. This is all obtained by a quantitative PCR analysis. We also found an association between grade 3 or 4 neurologic toxicity and peak CAR T-cell levels, with the patients with higher CAR T-cell levels being more prone to have severe neurologic toxicity. We found some very interesting cytokine correlates. This is given in much more detail in the paper in JCO that's available online. But IL-15 was the most important cytokine. We studied it many different ways and found statistically significant associations between response and IL-15 levels, and also between IL-15 levels and CAR T-cell peak levels. You can see that IL-15 peaks. The peak occurs about 2 days median after the CAR T-cell infusion. And patients with complete remission or partial remission had much higher peak levels of IL-15 than did patients with stable disease or progressive disease. Also, IL-10 had nice high peaks in patients with complete remissions or partial remissions, and much lower levels in patients with stable disease or progressive disease. So in summary, with this work, with the low-dose chemotherapy regimen, it certainly can cause lasting remissions of advanced lymphoma. The dominant toxicities were neurologic. This same CAR is under industry development by Kite Pharma. We have had some results published. Nilapu and Locke presented abstracts at ASH and ASBMT most recently in 2017, and Locke et al. Published in Molecular Therapy in 2017 some results from the multi-center trials of the same CAR, produced by a slightly different manufacturing process, but very similar to the data that I've shown before. The anti-CD19 CAR T-cells, I think, will probably become a very important therapy for B-cell lymphoma in the near future. Mainly because a lot of the patients that we treat really don't have any other good treatment options, and the CAR T-cells are effective. There's certainly a big demand among patients and referring physicians for CAR T-cell therapy for advanced lymphoma. But despite these promising results, many improvements are still needed. Improvements in the efficacy and toxicity are needed. Obviously, not all the patients got complete remissions, and there is substantial toxicity that needs to be improved upon. One thing I want to go over next, I think, is one of the important parts of this work is the long-term results. So we've been treating patients since 2009, and some of these patients had very long remissions. These are patients that are on a different cohort of the trial than I've discussed so far. These patients received a somewhat higher dose of cyclophosphamide plus fludarabine chemotherapy followed by CAR T-cells, and it's the same CAR as I talked about before. We had a cohort of 7 evaluable patients with diffuse large B-cell lymphoma. 5 of these 7 obtained complete remissions. The interesting thing about this, which I think is very important to the field and very important to patients and physicians, for these patients, so 4 out of the 5 that got complete remissions have had very long durations of their remissions, 56 months, 44 months, 38 months, and 42 months. So these patients have not had a relapse of their lymphoma, and this is, I think, really important because it shows that this therapy has the potential to give long-lasting remissions, which is exactly what all the patients want. And after the first 2 to 4 weeks or so, their toxicity goes away, so they're basically living normal lives for years after this therapy in some cases. This is some interesting correlative study from these 4 patients. We have B-cell recovery. Now, this is B-cell recovery out to the last follow-up. So these 4 patients had long remissions. One of them did not recover her B-cells. You can see the B-cell count 0 down here, all the way down on the 0 point, the open circles. The other 3 did recover their B-cells. So the dashed line shows the lower limit of normal B-cell count. These patients all recovered their B-cell count to above normal levels at certain points after their follow-up. This is all in months. So roughly anywhere from, these recovered around 9 months after, and this patient got up to normal about 22 months or so. But after they recovered their B-cells, their remissions have continued. So at the end of these lines, these patients are all still in remission, but normal B-cells recovered, malignant lymphoma B-cells did not recur. So I think that's really important. Another interesting finding, which I don't have an explanation for yet, is some of the patients that recovered their B-cell counts did not recover normal IgA levels in their serum. This is an example showing that the B-cells that recover in these patients are normal B cells, at least as far as we can tell. We have CD19 and CD20 expression by these B cells. This is one example. This is the patient with these solid triangles here. Polyclonal is shown by kappa lambda staining, and a normal cohort of naive B cells is shown by the IgD positive, CD27 negative population here. So as I mentioned before, despite successes, there's much room for improvement. So many patients do not obtain durable remissions. Toxicity is a major problem. I mentioned the 55% neurotoxicity in our last major report. And another problem is the possibility of recipient anti-CAR immune responses, which has been very nicely studied by Turtle et al. From Seattle. So to address some of these issues with the CAR T cells, I've been designing some new CAR T cells recently. So this is now showing some current work that is not published yet to try to improve on CAR T cells. So the previous CAR was designated FMC6328Z, the one that we've been using since 2009. So now I'm going to start talking about some newer CARs. One improvement is they use a fully human variable region, hopefully to decrease immunogenicity. And we've also been studying different co-stimulatory hinge and transmembrane domains. So I'm going to show just a very brief bit about this preclinical work in the interest of time, but this is unpublished preclinical work. We have made some changes in two hypotheses. One, changes in the amino acid sequence of the hinge and transmembrane region can affect the function of CAR T cells. And number two, a CAR with fully human variable regions will reduce recipient anti-CAR immune responses and perhaps increase T cell persistence. So we studied two CARs. One of them is called HU19CD828Z, has a human single-chain FV, a CD8 alpha hinge and transmembrane, CD28 co-stimulatory domain, CD3 zeta T cell activation domain. And the other one's called HU1928Z, and it's identical except it has the CD28 extracellular transmembrane and co-stimulatory. So all those parts are from CD28. This is, as I said, we have a lot more data than this, but in the interest of time, I really don't have time to show a lot. I'm going to show the most important, pertinent clinically thing that I think is the most important right now, that the CAR with a CD8 hinge and transmembrane domain consistently makes a lot less cytokine than a CAR with a CD28 hinge and transmembrane domain. So these CARs, HU19CD828Z and HU1928Z, are the same except for the difference in the hinge and transmembrane domain, with the CD8 hinge and transmembrane domain leading to lower levels of cytokine production. And here we're showing interferon gamma, TNF, and IL-2. We've shown nearly identical results with a different CAR and with many different types of target cells. So that's not all the preclinical work we have, but I'm going to go on to show a clinical trial that we just started recently with a fully human CAR showing the HU19 CD828Z. We're using now a third-generation lentiviral vector. I mentioned the fully human single-chain FV here, the hinge and transmembrane domain from CD8, and the CD28 co-stimulatory and CD28 co-stimulatory and CD3 zeta T cell activation domains. It's a dose escalation trial. We use the same cell culture process as before, stimulating whole PBMC with the anti-CD3 monoclonal antibody. Cell production takes 7 days. It's a dose escalation study, enrolling patients on these 4 dose levels. We're on dose level 3 now. We're going to treat advanced lymphoma again. We're focusing on lymphoma in the B cell space. We're interested in repeat infusions. Adverse events that we've seen so far in 12 patients. There has been some CRS, but it's been fairly mild to moderate. 1 patient did have grade 4 hypotension. 6 patients had grade 3 hypotension. Only 1 patient had grade 4 neurologic toxicity, and 1 had grade 2 neurologic toxicity. So in contrast to previous work, the neurological toxicity seemed to be less with this CAR compared to the other CAR, at least so far. So with this new CAR, with the various changes we made, 1 out of 12 neurologic toxicities compared to 12 out of 22 neurologic toxicities with the other CAR. 1 patient also had delayed grade 4 neutropenia, which is another toxicity we need to watch out for with CARs. Sometimes patients do have delayed cytopenias, which is another area of investigation. This is an example of a response, I think. Yeah, that's fine. This is an example of a response. The yellow area is lymphoma. The lymphoma disappeared after a month for the most part. There was a little bit of residual lymphoma here and then disappeared completely after 4 months. We have conclusions from this part of the study. Overall, in these 12 patients, we had an overall response rate of 75%, complete remissions in 50%, 6 out of 12. Only 1 of 12 had grade 4 neurologic toxicity in contrast to grade 3 or 4 neurologic toxicity on our previous trial. And results with the human CD828Z are very preliminary but promising so far. So now part 2 is the development of CAR T-cell therapies for multiple myeloma. We have done a lot of work targeting an antigen called BCMA, B-cell maturation antigen. This is known as CD269. It's expressed at least in low levels on the myeloma cell surface by almost all cases of multiple myeloma. We found that BCMA is not expressed on normal essential tissues, and we designed and performed preclinical tests on the first anti-BCMA CAR. This was published by Carpenter et al. In Clinical Cancer Research 2013. This is just one bit of the lab work that we did. Again, 2013 Clinical Cancer Research. We did a lot of other work leading up to this, but the mouse studies really encouraged us to go on and continue this work. We established solid tumors in mice with the BCMA positive tumor RPMA-8226 in NSG mice, as you know, very immunocompromised mice. So we established a human tumor cell line, then we treated the mice with human T cells. The anti-BCMA CAR eradicated the tumors, while the control CAR SP6 in the untreated mice just developed progressively enlarging tumors and went on to die. The CAR-treated mice with the anti-BCMA CAR survived long term. So we used this vector for the clinical trial. It's a gamma retroviral vector. We've treated now 18 patients. The CAR T-cell infusions are preceded by low-dose cyclophosphamide, as I showed in my previous anti-CD19 work. We've treated patients on 4 dose levels now. The first 3 dose levels, there was minimal toxicity and only 2 anti-myeloma responses. And this is a summary of those patients that received low doses. You can see we started with a dose of 0.3 million cells per kilogram, a very low dose. Then we went to 1 million per kilogram, then 3 million per kilogram. It's a little bit out of order. Patient 12 was treated at a lower dose than patients 10 and 11. But you can see that the response is a PR that lasted only 2 weeks and a very good PR that lasted 8 weeks. Most of the patients just had stable disease, so there was not much evidence of activity. There was also very mild or no toxicity in all the patients. We went on to increase the dose up to 9 million CAR T cells per kilogram, and when we did that, we found a much more robust activity, both with responses and toxicity. So we have here 7 patients who received this dose. And you can see every patient but one achieved either a very good PR or a stringent complete remission. Very good PR in the multiple myeloma staging system means that the myeloma burden was reduced by 90% or more. So the very good PR meaning a very good response. You can see the stringent CR, very good PRs. Some of these are ongoing. We have 3 ongoing. Some of them have progressed. Our longest response to date was a very good PR that lasted 66 weeks, and then the patient did develop progressive multiple myeloma. So the patients also experienced, well, first of all, we'll show a very representative example of a patient who had very high disease burden with a lot of multiple myeloma who received this 9 million CAR T cell per kilogram dose. He'd received 5 prior lines of therapy. He had severe cytokine release syndrome. All of his toxicities resolved completely, and his markers of myeloma decreased rapidly. So CAR T cells were infused on day 0. His temperature within 4 to 6 hours went up to approximately 41 degrees. So this is what we see often with the multiple myeloma patients with substantial disease burden. Toxicity comes on very quickly, at least with this CAR in this trial. The temperature stays up for a few days, then goes down. C-reactive protein, another measure of inflammation, also goes up to high levels. Along with this, we see now a big decrease in the M protein. The M protein is a monoclonal protein that's produced by the multiple myeloma cell, so it's a marker of disease burden. Also, the free light chains or monoclonal light chains is another marker of disease burden. Before treatment, with day 0 being the day of treatment, the patient had high levels of these markers, and they dramatically dropped to very low levels. And you can see they stayed down for about 400 days, and then progression occurred. This is an example. This is the same patient's bone marrow before treatment. The pink is CD138 positive plasma cells. Brown is BCMA staining of the bone marrow. So the patient had a lot of myeloma. 80% of his bone marrow was myeloma before treatment. 4 weeks later, his bone marrow was hypocellular with no evidence of plasma cells at all. Notice the pink is all gone. 8 weeks after, his normal hematopoiesis was returning, but the plasma cells were still completely absent. So we eradicated the plasma cells in this patient, and they continued to be eradicated for several months. This is an example. This is CAR T-cell levels in the blood of these patients. Patients with the low CAR T-cell doses had very low levels of CAR T-cells. The one patient with a nice peak of CAR T-cells was the only patient in this group that had a very good PR or better. The patients that got the higher dose of 9 million CAR T-cells have much higher levels of CAR T-cells in their blood. BCMA is a serum protein, so this work shows that CAR T-cells can function in an environment where the serum protein, where the protein being targeted is in the serum. So despite serum levels of BCMA, patients still could get responses. So these patients, 10, 11, 13, 14, 15, 16, had responses, and BCMA dropped. So with response, BCMA dropped, and BCMA did not preclude responses. In contrast, patients that did not have responses were all straight lines. Their BCMA serum levels didn't change hardly at all, except we had this one patient here who got a very good PR who did drop. So this is the patients on the low dose. The only patient who had a drop in serum BCMA was the one patient who had a very good PR. So disease burden was associated with serum BCMA levels. One potential problem with this approach, especially in multiple myeloma, which is a heterogeneous disease, is loss of the antigen. So we have shown in one patient very clearly, I think, patient 7, loss of the BCMA antigen on the surface of her myeloma cells. This patient had uniform BCMA expression as shown by the red population of cells here. These are the plasma cells that are BCMA positive with very few or very, very few BCMA negative plasma cells before her treatment. Then 2 months after treatment, there was quite a big population of BCMA negative cells in this patient. The purple is bone marrow CD19 positive cells, which are included as a negative control for BCMA staining, because most B cells are actually BCMA negative. I think the variety of cytokines elevated during the cytokine release syndrome after CAR T-cell therapy is a very important area of study. These are 2 patients that had responses on the BCMA CAR trial. We measured serum cytokines in their blood at the time of their toxicity. And you can see a wide variety of cytokines were elevated. This is given as fold increase in the cytokines over baseline. So you can see interferon gamma, IL-10, a variety of other cytokines, IL-13, interleukin-4, GM-CSF, IL-15 was elevated, IL-17A, IL-6. So a wide variety of cytokines are elevated in the patients after CAR T-cell infusion. I think this is an important thing to remember and an important area for future study to try to figure out if other cytokines, aside from IL-6, could be blocked to help treat the toxicity. So most people are aware of tocilizumab, which is an IL-6 receptor blocker that's used to help abrogate IL-6's impact on toxicity, but there are perhaps other opportunities here to help treat toxicity without hurting responses. So the future plans I have, first of all, we demonstrated that anti-BCMA CAR T-cells have powerful activity against multiple myeloma. Toxicity caused by a variety of cytokines is a limiting factor for many different types of CAR T-cell therapies, for both the anti-CD19 in many cases and also the BCMA and other types of CAR T-cell therapies. I think almost all the toxicity that's caused by CAR T-cells is caused by cytokines and other types of soluble substances released by the T-cells. The evidence for that is that the toxicity is so short-lived. Usually the T cells go in, the patients have severe toxicity that lasts anywhere from just a few days up to a couple weeks, and then the toxicity usually goes away very quickly. So I think that's evidence that the toxicity is caused by some soluble substance that goes away. We don't really see a lot of direct damage. We haven't seen really with my studies any direct damage to organs of the CAR T cells reacting against organs and hurting the organs directly, but it's the toxicity that I think mainly caused by soluble substances from the T cells. As I mentioned, it's an important area for future research. Also, especially for multiple myeloma, targeting different antigens is critical. Multiple myeloma is a heterogeneous disease. Despite its clonal origins, it branches out over time, and there's phenotypic differences between multiple myeloma cells in the same patient, so it's really critical to find new antigens for multiple myeloma especially. So thank you all for your attention. Now, the acknowledgements. My current branch, ETIB of NCI, my branch chief is Ronald Gress. A close co-worker, Jennifer Brudno, does a lot of the clinical work that I've presented. Many other co-workers in my lab and in the branch and clinical areas. Kite Pharma has been a good collaborator with the CD19 work. Surgery branch, Stephen Rosenberg, I trained in his lab, and he definitely got me started in the CAR field and was a very close collaborator in the CD19 work, especially in the earlier work up until just a couple years ago. Really, we're still close collaborators now, but he was very instrumental in the early CD19 work. Other collaborators at the NCI and other areas of the NIH. Thank you.
Thank you. Thank you, James. That was really wonderful work. The floor is open for questions for James. So actually, I had a question regarding the heterogeneity of the multiple myeloma cells. Is it, if you did these as a frontline therapy versus as a last sort of therapy, do you think that that would impact the heterogeneity? I mean, do you think that the chemotherapy is contributing to the heterogeneity?
I think it's quite possible that starting earlier would have a more homogeneous multiple myeloma population. So I think in CAR T cells in general, I think they probably will move earlier in treatment. I think probably the first CAR T cell product to move earlier will be the anti-CD19 CAR T cell therapies for lymphoma. Certainly, it is possible that this branching of the phenotype of multiple myeloma could be minimized by treating earlier.
Yes, for the BCMA trial, there's no neurological toxicity.
So far, we have had delirium in a few patients. So when patients get really sick and they're in an intensive care unit and they're highly febrile and they have low blood pressure and various other problems, they can have some delirium. We've had 2 or 3 patients with delirium. But the toxicity, we have seen nothing like the anti-CD19 experience where we have the various types of tremors and ataxia and aphasia and the various other types of neurologic toxicity that we've seen with the anti-CD19 CAR. Yes, go ahead.
Murad Hassani from NIH, Office of Science Policy. On some of the BCMA CAR trials, they have seen some elevated CPKs that correlated with muscle weakness. Have you seen that in your trial?
We saw that in one patient. Patient number 10 did have an elevated CPK. That's not specific for BCMA, though, because I've seen that in anti-CD19 CAR patients, too. I've seen that in patients with ALL who had severe cytokine storm with the anti-CD19 CAR. We've published that in JCO 2016, Brudno et al. I didn't talk about it today. We had a trial of allogeneic anti-CD19 CAR T cells for patients that relapsed post-transplant. We treated some ALL patients, and one of those patients at least, maybe 2 had, no, at least 2 of them had some elevated CPK and muscle pain and weakness. On the BCMA trial, again, a patient with really severe cytokine release syndrome, we had at least one patient who had some muscle weakness and pain and elevated CPK.
And just a last question, considering some of your new constructs, including the humanized construct with CD19, and seeing less neurotoxicity, do you think that some of the neurotoxicity seen on the original CD19 trials may be related to the construct?
I think My theory with the CD19 is that with the neurotoxicity, obviously nobody really knows, but I think a lot of the neurotoxicity is caused by soluble substances, just like everything else. There's something secreted by the T cells that causes this neurological toxicity because I have no proof of that, but we do have evidence that the T cells certainly traffic to the CSF. We published that as in the JCO 2017 paper and a little bit in the JCO 2015 paper. But we have detected some elevated cytokines in the CSF, but nothing consistent to say this goes up. Other people have published cytokines. Cameron Turtle published some things on neurologic toxicity in his Science Translational Medicine paper in 2016. But just the clinical picture of the patients having neurologic toxicity that only lasts a few days and then goes away in most cases tells me that it's probably some soluble substance that causes toxicity and then dissipates, as opposed to a direct damage to the brain tissue, because I think if the brain tissue was being directly bound to and damaged by the CAR T cells, the toxicity would not go away. There would be some residual toxicity left. And at least in my patients, we haven't had any long-lasting neurologic toxicity.
Great. One last question.
In your immune recovery post-CD19 CAR T, have you looked at response to vaccination?
No, we haven't. That's an interesting area of study, but we haven't really studied the patients by vaccinating them to see what happens.
So I'd like to move on to our second awardee, Lily Yang, who comes to us from UCLA. And Lily's achievements that really stood out among the pack was her work on directed differentiation of hematopoietic stem cells to become invariant natural killer T cells. And she did this by introducing into the hematopoietic stem cells genes encoding T cell receptors cloned from these invariant cells. And in this manner, this work is described as to be Harry Potter-esque. The wand is choosing the wizard. And her work is also, she also has extensive work on expanding the capacity of xenograft mouse models for studying human hematopoiesis and lymphopoiesis that I think is going to yield new discoveries in the future. And finally, she is working on the fundamental exploration of lymphocyte function with a particular focus on the roles of non-coding RNAs, microRNAs, in modulating lymphocyte responses. So her work is really very broad and very exciting, and we're delighted that she was nominated. And so with that, I'd like to congratulate Lily on receiving this award. And now she's going to present to us her work.
May I have my slide on? Okay. Thank you, Dr. Davidson, for this very kind introduction. Here's my disclosure. I would like to start my talk by thanking my two nominators, Dr. David Baltimore and Dr. Donald Cohn. Dr. Baltimore is my PhD advisor and also my postdoc advisor, who guided me into the research field of immunology and has been a strong supporter for me throughout my career development. Dr. Donald Cohn is my faculty mentor at UCLA, who has collaborated with me and guided my research since I joined UCLA. Dr. Cohn is in the audience today. Thank you, Don. I would like to extend my acknowledgement to the Engineering Immunity Consortium. This consortium gathered the clinicians and research scientists at the Los Angeles area who are interested in cancer immunotherapy. We gather regularly to exchange ideas, to form collaborations, and to promote the translation of the new kind of immunotherapy. The consortium comprises over 30 research laboratories and clinical teams from the local institution, including UCLA, Caltech, USC, and the City of Hope. Without their support, I will not be able to accomplish the work I'm going to share with you today. My research centered on this concept called engineering immunity. We aim to genetically modify the various components of the immune system, in particular, the various types of immune cells, to enhance immune responsiveness against target diseases. Among the many engineering immunity approaches that I have explored, I would like to highlight 3. At one direction, we tried to engineer dendritic cell immunity against cancer and HIV/AIDS. Dendritic cells are considered to be the most potent stimulator of T cell and B cells in vivo. We have constructed lentiviral vector that has been enveloped with engineered receptors. The receptor can specifically recognize dendritic cell surface marker called DC sign. The vector can encode the antigen that either target infectious disease like HIV or target cancer like encoding tumor antigen. The engineered lentivector, upon directly injected in vivo, can specifically transduce dendritic cells, deliver the antigen gene into dendritic cells. The dendritic cells then encode the antigen and stimulate T cell and B cell response. This direction of research resulted in the listed publications. The technology is also licensed by a technology company called Immune Design for commercial development. At another direction, we tried to engineer B cell antibody immunity against HIV/AIDS. Neutralizing antibody are considered to be promising reagents to block HIV infection. We cloned out the neutralizing antibody gene, either using lentiviral vector to deliver the gene into hematopoietic stem cells that will result in B cells secreting neutralizing antibody, or we use AAV vector to encode the neutralizing antibody genes and deliver the gene into muscle cell and makes the muscle cell to produce neutralizing antibody. For the AAV approach, through direct intramuscular injection, the transduced muscle cell will constantly secreting large amount of the anti-HIV neutralizing antibodies and block the HIV infection. This direction of research results in the listed publications, and the technology is under clinical development. Today, I would like to focus on talking about the third direction, that we tried to engineer T cell immunity against cancer. From last lecture, Dr. Kochendorfer gave us a very nice introduction about cancer immunotherapy, which aims to enhance the immune response and utilize the power within our body to battle cancer. It's considered to be a major breakthrough in modern medicine for cancer therapy. From the previous lecture, we also learned that the T cells are potent therapeutic target for treating cancer. A few facts about the T cells. Like all the other immune cells, T cells derived from the ultimate progenitor cell called hematopoietic stem cells. Those stem cells live in the bone marrow of adult human. For T cell development, the progenitor cell migrates to an organ called thymus, which is located above the heart. In thymus, T cell will complete the development. The mature T cell then exit thymus and then go to the periphery and ready to protect us. There are 3 fundamental rules that govern the T cell immunity. First, population diversity. Each single T cells express a different kind of T cell receptor. Taken together, the T cell repertoire have a very diverse range to cover and protect us from all the possible pathogens that may invade our body. Second, clonal specificity. As mentioned, each T cell expresses one clonotype of T cell receptor and allow them to have a designed specificity. Third is a clonal expansion. Upon recognition a specific antigen, the T cell clones that recognize this specific antigen will go clonal expansion, duplicate themselves to become many T cells of the same specificity, recognize the same antigen. This mechanism works very well and protects us against the majority of the infectious disease. However, the T cells often lose battle against cancer because of two major reasons. One is because for most tumor antigen, they are considered tumor cell derived from self. They are considered a kind of partially self. So the developmental mechanism regulation limit the number of T cells that can exist for targeting cancer. Another reason is that the tumor environment oftentimes produce a inhibitory signals that inhibit the expansion of the clonal T cells that due to this tumor suppression mechanism. So to utilize the power of T cells to tackle cancer, adoptive T cell therapy has been developed. For this approach, either the tumor-infiltrating lymphocyte has been isolated and expanded in vitro to move them away from the tumor inhibitory microenvironment, and expand to large numbers in vitro, then give back to the patient to increase the number of anti-cancer T cells. Another approach is gene-engineered peripheral blood T cells. Those T cells can be engineered to either express the anti-cancer T cell receptor to redirect the specificity against cancer, or they're engineered with a chimeric antigen receptor to target them to tumor surface antigen. From the previous talk, we have a nice introduction about the impressive achievement on utilizing the CAR T therapy to attack the blood cancers. A major challenge for the adoptive T cell therapy is to achieve the long-term persistence of the anti-cancer T cells in vivo post the adoptive transfer. We tackle the challenge through a different angle. We go back to the ultimate progenitor cells of the T cells, the hematopoietic stem cells. Our strategy is to clone out the anti-cancer T cell receptor and deliver the gene into the hematopoietic stem cells. We utilize two mechanisms that guide the T cell development. One is called allelic exclusion. In this mechanism, once the progenitor cells during the T cell development sense the presence of a pre-rearranged T cell receptor, which is a transgene we put in, it will stop the recombination of the endogenous T cell receptor. So the resulting T cells coming from these engineered stem cells will carry the single specificity we designed them to carry, which is to recognize the cancer antigen. Another mechanism we take advantage of called the TCR instruction mechanism. This mechanism allows the specific T cell receptor to guide the development of a special subtype of T cells. For instance, if we put a CD4 helper T cell receptor into the hematopoietic stem cell, we will guide the stem cell to develop into helper T cells. If we put a CD8 T cell receptor into the stem cells, we'll guide the development into cytotoxic T cells. By doing this, we are able to program the hematopoietic stem cell to produce the tumor-targeting helper or cytotoxic T cells. This approach, we have a couple of advantages. Because of the longevity and self-renewal of the hematopoietic stem cells, once given to a patient, the engineered stem cell will guarantee the providing to patient a lifelong supply of cancer-fighting T cells. During my PhD study, we proved the principle of this method using mouse model. Our collaborators at UCLA, the Zach lab and the Cohn lab, then proved the concept in humanized mouse model to show that this also works for the human stem cells to generate the human and cancer T cells. Clinical trials are ongoing currently at UCLA Medical Center, led by Dr. Anthony Ribas, testing the HSC T therapy for treating multiple cancers. In 2013, I moved to UCLA, started my independent lab. In my lab, we developed a new interest. We're now interested in this special type of cells called invariant natural killer T cells, iNKT cells. What are those cells? Why we are interested in them? Consider a drop of human blood. It contains about 10 million of the total blood cells. Among those, about 0.1 million are white blood cells. Those are the immune cells. Among the immune cells, we have about 5,000 conventional T cells, and we have about 10 iNKT cells. So those cells are very rare. However, despite their small number, iNKT cells are very powerful cells to tackle cancer. In the war against cancer, if we consider the conventional T cells to be the foot soldiers, the iNKT cells can be considered as special forces. They respond stronger and faster. iNKT cells are a unique type of T cells, very different from the conventional T cells. Unlike conventional cells, they do not recognize classical MHC class 1 or class 2 molecule. They recognize a non-classical class 1 molecule called CD1D. They also do not recognize peptide antigen. They recognize glycolipid antigen. So that gives them the special capacity to recognize a totally new spectrum of the lipid antigen that may be presented by cancer cells. There are 3 mechanisms have been proposed that how iNKT cell may attack cancer. For the tumor cells that express CD1D, iNKT cells can recognize the CD1D displayed lipid antigen and directly attack the cancer cell. In addition to that, they also are very potent stimulator of the NK cells. They can stimulate NK cell to attack cancer. For the CD1D negative tumor, iNKT cells can indirectly be stimulated through recognizing the APC, antigen-presenting cell, presented CD1D and the lipid antigen, get stimulated, stimulate NK cells, which then attack tumor cells. iNKT cell can also inhibit the tumor-associated macrophage, which contribute to the suppressive environment inside of the tumor. By suppressing the tumor-associated macrophages, iNKT cell can stimulate NK cell to attack tumor and also change the environment to help boost the anti-tumor immunity. Because iNKT cells recognize CD1D molecule, which is non-polymorphic, and also it recognizes it can direct or indirectly recognize tumor cells. They are considered to be a pretty common, cell reagent that can attack multiple type of a tumor independent of MHC and tumor antigen restriction. Because of the properties we just talked about, iNKT cells, they are attractive cellular agent for cancer immunotherapy, but the clinical application is greatly limited by the low number and high variability in humans. We just talk about those cells of low number, but within the human population, there's a very high, big range. Like for the people sitting in the audience today, if we compare our iNKT cell number inside of our blood, it can range from 0.001 to 1%. For cancer patients, usually their number of iNKT cells are at the very low end. And within cancer patients, the patient have relatively higher level of iNKT cell in many types of cancer correlated with better prognosis. So we propose that HSC engineered iNKT cell therapy may provide a new opportunity to really harness the power of this promising type of cells to utilize them for attacking cancer. Our strategy is to clone out of the iNKT T-cell receptor gene, use lentiviral vector to deliver those genes into hematopoietic stem cell, then program those stem cells to develop into iNKT cells. Those iNKT cells now can be provided for long term and for constant level, and hopefully we can bring it up to above the therapeutic level that the patient can utilize those cells attacking their cancer. For proof of principle, we use the mouse model. We clone out the mouse iNKT cells using the viral vector delivered into the mouse bone marrow stem cells, and through a common bone marrow transfer approach, transfer those cells into recipient mice. Then we track the mice to see whether we can generate iNKT cells. So I want to bring your attention over here. So those are the control mice. Those are the engineered mice. The engineered mice, those show the fold change. We are able to increase the iNKT cells with a very high fold change. Because the bone marrow transfer approach, we can adjust the ratio of the T-cell engineered stem cell versus the non-engineered cell. We can easily titrate down the number and control the output to generate the level of iNKT cells that fit our need. The stem cell engineered iNKT cells, if we look at their phenotype, they express the transgenic T-cell receptor. They co-express the NK cell marker. They display a memory phenotype. They display the typical CD4 and CD8 co-receptor expression. Overall, they look very much like the typical iNKT cells. And we talked about the advantage of stem cell engineering is because of the longevity and the self-renewal. In our engineered animals, we track the animal for as long as the experiment goes, for up to 6 months, that the mice continue to produce high level of engineered iNKT cells. We even tried the secondary bone marrow transfer to really test the longevity of the engineered stem cells, and we found through secondary bone marrow transfer the high level of iNKT cells maintained. Of course, most importantly is to ask whether the stem cell engineered iNKT cells are able to mount anti-cancer immunity. Here we utilized a pretty malignant melanoma lung metastasis model, the B16 tumor model. We inject the tumor cells into the mice. I want to bring your attention to be here. So for the control mice, those tumor cell metastases very quickly in the lung form many tumor nodules over there. For the mice that are engineered with our iNKT cells, those mice are greatly protected, and we can see a large reduction of the tumor nodule. We can also boost the iNKT cell response through immunizing the mice with a known antagonist antigen called alpha-galther that specifically stimulates iNKT cells. Through the immunization, we can see a further protection and a reduction of the tumor nodule. I would like to bring your attention to be here. So you may notice that in the mice that are giving iNKT cell therapy, we see reduced number of nodules, and also majority of the nodules are colorless. The loss of pigment is a very common phenomenon people observe for the melanoma patient when the melanoma cancer are under severe immune attack, because a couple of key enzymes involved in the synthesis of the pigment are very well characterized melanoma tumor antigen that can stimulate immune reaction. So loss of the color means that the tumor are forced to change those key enzymes. So it's a nice evidence to show that engineered iNKT cells are able to induce strong anti-tumor immunity. To summarize what we just talked about, the HSC iNKT cell therapy is a new concept of cancer immunotherapy. We already showed the proof of principle in mouse tumor model. Now I'm going to talk about our ongoing research. So next step, we want to really promote the translation of this new therapy. Last year, we received support from the California Institute for Regenerative Medicine, support the translation of this HSC iNKT therapy into clinics. We formed a large translational team to promote this work. So it's comprising 15 UCLA researchers listed over here. Majority belong to the Engineering Immunity Consortium I mentioned to you at the beginning of the talk, and also have a 3 external board. This is our proposed therapy. For the cancer patient, the patient will get treated with GCSF that will mobilize the hematopoietic stem cells in human, which are the CD34 positive cells. GCSF will mobilize those stem cells from bone marrow, move to the peripheral blood. Then through the leukapheresis, we can collect those CD34 cells. Those cells will be put in vitro in culture in contact with a lentiviral vector delivering the iNKT T-cell receptor. The transduced stem cells will be infused back into the patient. In the patient, those cells will localize to the bone marrow and will go through the T-cell development. Some of the progenitor cells the engineered cell will then be programmed to develop into iNKT cells and produce a constant supply of the iNKT cells for the patient. For the translational purpose, we are utilizing a humanized mouse model called the BLT model. It stands for human bone marrow liver thymus reconstituted NSG mice. Those are immunocompromised mice will take implant of the human hematopoietic stem cells. We will also give the mice a piece of human thymus to support the T-cell development. After the reconstitution, the mice will be allowed some time for the T-cell to develop, and then we will examine the presence of the human iNKT cells. The mice will also receive the human tumor xenograft that will allow us to examine the anti-tumor capacity of the stem cell engineered iNKT cells. Here is what we have achieved. We have we sought out the human iNKT cells from peripheral blood, clone out the iNKT T-cell receptor genes. We constructed a lentiviral vector. we've also put a GFP marker together with a TCR gene for convenient monitoring of the engineered cells. This vector used to transduce human CD34 positive cells, then given to the BLT humanized mice, and then we track the presence of the human iNKT cells. Here shows that after 6.5 weeks post the stem cell transplantation, if we look at the blood of the humanized mice, we are able to detect the human blood cells. If we look at it into the human blood cells, we see the GFP positive cell, which indicate those cells come from the genetically modified stem cells. If we look into the GFP positive human cells, we notice that the majority of those cells are human iNKT cells. As a control, the non-transduced human cells, we do not, the iNKT cell number is below detection level. So this indicates that the T cell receptor gene we gave to the hematopoietic stem cell successfully programmed the human stem cells to generate human iNKT cells. So currently, we're testing this new therapy against various human cancers in humanized mouse model, and we're also promoting the translation of this new cancer immunotherapy into clinics. We aim to finish a pre-IND meeting with FDA by the end of year 2018, and hopefully by that time, we can move on to clinical trial. I would like to closing by thanking my lab people who have done the work, in particular, my first PhD student, Drake Smith, who has been leading this project. I would like to thank my collaborators and also my funding support, in particular the CERN grant that allow us to translating the HSC iNKT therapy. I'll stop here. Thank you for your attention. I'll be happy to answer any questions you may have.
We have time for a couple questions. Right here.
That's a very nice talk.
Thank you.
I have two questions. So if you want to move the model from mouse to human, my question is, first you probably want to do, so during the stem cell transfer, you want to do busulfan depletion of the bone marrow, right?
Yes.
And then during that time, the patient had like whatever cancer, and then probably because of the busulfan depletion of stem cell, it probably take a while for the bone marrow that you transplant in there to fully grow and then reconstitute the patient's immunity. During that time, do you think it's going to be a problem that for the patient doesn't have enough immune cells to control the tumor before the NK cells to come out?
That's a very good point, right? So for the previous clinical trial, the HSC T cell clinical trial, which will generate the conventional T cells. So the current clinical protocol is during the gap after the stem cell transfer and before the new T cells come out from the stem cell development. During that window, that we are going to give the patient the preferred blood T cells engineered to express the anti-cancer T cell receptor. It's the same T cell receptor we gave to the stem cells. But for the peripheral T cells, once engineered, they are able to cover that period of time. So it's combined.
Your cells were derived from the 0.1% to 1% of the peripheral blood cells?
So that are the conventional T cell therapy. For this new therapy, what we propose is that we can collect some peripheral blood cell from the patient before giving them, chemotherapy, precondition, and for stem cell therapy. So we get the peripheral blood cell that will contain the iNKT cells. We can stimulate the iNKT cells in vitro, grow them to a certain number, and as a mixture, so it will be autologous transfer of the patient PBMC cell enriched or promoted for the expanding of iNKT cells that can give back to the patient. So again, cover the period of time that the patient is waiting for the T cells coming out from the stem cells.
You need to move on over here.
Yes, I would like to ask you a question about your mouse model. So, have you ever attempted treatment of large established B16 melanoma in your model?
Yeah,
Because you are showing
the
pretty much
prevention.
protection rather than therapy. And you know, in the clinical trial, I assume you will aim to treat people with metastatic disease. So.
Good question. Yeah. Again, so for the mouse model, we're limited by the timing. It takes about 8 weeks for the stem cell approach to work to allow the engineered iNKT cells to derive, and all the existing xenograft tumor model doesn't really last that long. So we actually tried a alternative model. So we pre-grow the tumor in a mouse to form the pre-established solid tumor. Then we collect all the peripheral cells, like the spleen cells from an engineered mice, transfer that into tumor-bearing mice. We did find that the stem cell engineered iNKT cells will be able to control the established tumor for that. It's not exactly the approach that we foresee happening in patient, but for the limiting of the mouse model, that is what we can do. For the humanized mouse model, we plan, to do the similar thing. We plan to grow the xenograft tumor, human tumor, in NSG mice, and we'll collect whatever the peripheral, or the entire peripheral immune system generated for the BLT mice, engineered with iNKT, enriched for the iNKT cell. We'll transfer those cells to the tumor-bearing mice to mimic the condition that those cells coming out from the thymus and encounter a pre-established tumor. So that is a model we're trying right now.
So was it curative?
Well,
we're
really, sorry, we're running into the next session, so I want to give 2 more opportunities for questions, very quick questions,
later.
and 2 very quick answers, and so that we don't take up the next speaker's time. But this is a very exciting topic.
So I missed, what is the instructive signal you use for the? Maybe you said it, but I didn't catch it. How do you instruct the stem cells?
So everything, we didn't give anything extra. Everything is buried in that T-cell receptor sequence. So it turned out the particular T-cell receptor, once they display on the developing thymocytes, they will deliver the proper signal to guide the development to a special lineage.
Oh, I see. Okay.
Last question.
Thank you. Very nice talk.
Thank
Dr.
you.
Yang, do you have a glycolipid in mind that you want to use to go with the NKT cells?
Pardon?
The glycolipid?
Glycolipid.
Yes.
Yeah, so this is one tricky, question for the research field for iNKT cells, because they recognize glycolipid as not as easy to be studied as a protein antigen because it's not genomically, engineered. So we do have a, clinically actually applicable agonist called alpha-galactoceramide, alpha-galcer, that very specifically and potently stimulate iNKT cell. So that reagent we can utilize to boost the iNKT cell response.
So with that, I really want to thank our new investigator awardees, and again, help me congratulate them on their beautiful work.
Thank you.